Howdy, my name is Alex Thompson, and today I'm going to talk to you about how deformed wing virus, aka DWF, increases mortality in bees and elongates emergent time. So let's start off with the basics. What are honeybees? Honeybees, are otherwise known by their scientific name, Apis mellifera, are highly important for pollination in both commercial agriculture and for native wild and flowering plants. They can be exposed to pesticides and pests when foraging for pollen. Pesticide residue has been linked to health issues and colony failure. And to the right, in figure 1, you can see a picture of an Apis mellifera for yourself. And next, to introduce us to this topic, let's talk about DWV. That stands for deformed wing virus. It was first examined in the 1980s in Japan. It is an icosahedral virus, meaning that when it infects a cell or an organism and that organism or cell dies, the virus then can split open from that cell and spread that way and come out of the cell and infect other cells. And so it is pretty contagious. This virus is transmitted by the varroa destructor that is more commonly known as a varroa mite. They spread this disease and infect colonies all around the world. This disease is largely associated with colony failure, and so to the right in figure 2, you can really see why it's called deformed wing virus. You see this bee has these wings that are highly deformed. And let's link those topics together. How does DWV impact Apis mellifera and specifically their development? So this was an examined question by a University of Veterinary Medicine in Canada, and the way they set up this experiment, they left 5 hives untreated for mites, and they left them out all fall and all spring. After that, the 300 workers were then collected and the mites were removed and counted. And the queens in these 5 hives were then secluded for 24 hours to lay eggs. This was important so that they could know the exact time that these eggs were laid. The queen and the eggs were then placed back into the hive and placed next to a healthy hive, and they compared those eggs, the healthy eggs, to the infected eggs. And over the next few weeks, they observed and recorded the emergence time of every cell, the degree of deformity of the bees that emerged, and the survival rate of these bees. So in these 2 groups, it is important to note how many bees were infected. So in the group with the DWV impacted hive, 11% showed signs of infection, and all of the infected bees showed an protruded proboscis. So this is the proboscis here, and so on the left you have a healthy bee, on the right you have an infected bee. You have this proboscis, which acts kind of like a straw to suck up nutrients. And so this may indicate lower energy levels, and as you can see on the left, that didn't happen. I would also like to note that the infected group had a 30% higher mortality rate. And speaking of emergence, how long did it take for these bees to emerge from their cells? So as you can see, the normal group, you have, like, this is when the majority of these bees emerged. And here, with the deformed wing group, you have a lot larger spread. And as you can see here, on average, it took the deformed wing group a lot longer. I would also like to note that the normal group had 192 bees while the deformed group had 21. I think that does speak to the mortality and infection rate. So let's talk about other physical deformities. So here we have in figure 5, here on the left, this represents a healthy bee, and this represents a deformed bee. So the affected bees had smaller hypopharyngeal glands. That is, if you can see this little box here, and this box here, these two images are enlarged versions. So these green arrows right here, that represents the vesicles in the cell, and as you can see on a cellular level, the affected bees had very few of them, and they, if you look at like this gland versus this gland, as you can see, they were a lot smaller. And if you can't see, I will show you on the next slide. So here, this is the total area on average of the hypopharyngeal gland for the normal and deformed wings group there. And so what did I just tell you? In short, we know that DWV largely impacts Apis mellifera. It has been globally shown. Bees impacted with DWV, excuse me, have overall slower development, emergence from the cell, and higher mortality rates. The affected bees also have protruded proboscis, and smaller hypopharyngeal glands, which are crucial for brood development. Now, why is this significant? So, DWV is considered to be a very large threat to honeybee colonies. It has been detected in approximately 90% of French apiaries, and this virus has been detected in 100% of all mites that have been tested. More research is needed to determine information about both DWV and the exact biological interactions of DWV with Apis mellifera. Thanks, and Gigam.